welcome to today's webinar. Today we are discussing demystifying high-C data normalization. This is one of our Ask the Experts webinar series, and we encourage you in these webinars to bring your questions. It's an open discussion. It makes for a lively exchange between all of us, and certainly the questions that you ask will be undoubtedly questions that others have had. So we really encourage you to bring your questions to our experts that we have today. And let me just move on to introducing them. Today, we'll hear from Dr. Miriam L. Kawand. She's our head of customer success at Dovetail Genomics, as well as Dr. Jonathan Torchia. He's our senior bioinformatics scientist at Dovetail. Let me tell you a little more about Miriam. She leads customer success for proximity ligation kits at Dovetail, and she's been with the company since 2018. Miriam completed her PhD in molecular microbiology in the UK at the University of Warwick. And prior to joining Cantata, also known as Dovetail, she was a lecturer in molecular biology and genetics at St. Joseph University in Lebanon. She's going to be kicking us off today with an introduction to our topic, and then we'll hand it over to Dr. Torchia. He is a biologist turned bioinformatics scientist with over a decade's worth of experience with multi-omics analyses, and he received his PhD at the University of Toronto, where he studied the epigenetics of rare pediatric brain tumors. He followed up his PhD training with 5 years at the Ontario Institute for Cancer Research, where he led the initial development of CAP/CLIA-approved personalized oncogenomics, bioinformatics, pipeline, and translational research products. He continues his scientific curiosity to this day with Cantata Bio's R&D team, where he works on the development of the next iteration of 3D epigenomic tools and analysis workflows. So we're very pleased to have both these experts here to guide us through this very somewhat complicated topic, but they're going to make it easy and enjoyable for you to learn and to bring your questions. So with the questions, please use the Q&A box. We won't be monitoring the chat box, so please use the Q&A box at the bottom of your screen. Ask your questions at any time during the talk, and then once we have a break at the end, the two talks will be about 10 minutes each, so we'll have about 20 minutes of lecturing and then about 10 minutes for question time afterwards. So you can put them in the Q&A box at any time, and then we'll present them to the speakers at the end. You can watch for my email. I'll be sending out the recording and most likely the slides as well. So once the webinar is over and the recording has downloaded, then I'll send that out to you all so you can look for that email. That's going to come from me, Jessie Kennedy. All right, have a wonderful webinar, and I will pass it over to Miriam now. There she is.
Thank you, Jessie. Let me just share my screen. Can you see my slides?
Yes, we can.
Perfect. Well, thanks everyone for joining. In my part of the presentation, I'll speak about why there is a need to normalize 3D genomics data, and then I'll hand it over to my colleague, John, who will walk you through how to actually normalize the data. So there are two types of research goals from generating the 3D genomics data: discovery or comparison. So if your research goal is a discovery, you're really looking at studying the chromatin conformation of a single sample or experimental condition. So you're really looking at calling topological features such as AB compartments, TADs, or loops in your sample. And then you're likely going to add complementary data sets such as RNA-seq, ChIP-seq, ETAC-seq, to get a more holistic picture of what is going on in your sample, what's happening in your sample. For example, how a mechanism or a gene you're interested in is being regulated. If your research goal is more a comparison, you're looking at comparing that chromatin structure between two or more samples or experimental conditions. So here you're still doing a topological feature calling as a first step, but you're taking this one step further to compare the topological features between the samples. So your questions here are more around, has there been any switch from an A to B compartment between the samples? Has there been any disruption of a DAD structure between the samples? What are the loops that were gained? What are the loops that were lost? And what loops or interactions remain the same between these two samples? So regardless of whether you're doing a comparison or a discovery research goal with your data, you're actually going to set up your experiment also taking into account what is the topological feature that you're after. So if you're looking to call AB compartments, for example, you're looking at 100 kb resolution. That is usually attained with a 3x coverage of your genome that you can get to by 30 million read pairs, 2 by 150 base pair depth. And that's really talking about a human genome. And for you to generate that 30 million read pairs, you only need to generate one library, so one 3D genomics library that will support this depth. If you're looking at TADS, that's a little bit more resolution that you're really targeting. You're looking at a 10 to 25 kb resolution. So you need a 30x coverage minimum of your genome. This is achieved by sequencing to 300 million read pairs, 2 by 150 base pair. And it still can be achieved by generating only one library. So each library usually supports up to 300 million read pairs depth. Now, if you're looking at calling interactions or between your promoter and enhancer, for example, you're looking at loops, you're actually aiming for a higher resolution that's 5 to 10 kb. This is an 80x coverage of your genome that's minimally achieved with sequencing 800 million read pairs, 2 by 150 base pair. And for you to achieve that 80x coverage of those 800 million read pairs, you're looking at generating 3 libraries or 3 data sets. As I mentioned, each library will allow you to support up to 300 million read pairs. So you're going to generate 3 libraries, each sequence to 300 million read pairs, and collectively you're going to have that 800 million read pairs that you need to call loops at a 5 to 10 kb resolution. Now, there are different ways for generating these 3 libraries. You can do that by doing a technical replication, a biological replication, or a combination of both. And before I talk about the replications that you can design in your experiment, I just want to orient those of you in the audience who are not very familiar with the workflow. So the standard 3D genomics workflow that we offer has 5 stages. In stage 1, this is where you're preparing your sample. You're cross-linking the chromatin in the nucleus. Then you're digesting the chromatin with MNAs. You proceed to stage 2, where you check that the chromatin was properly digested. This is more of a QC checkpoint early on in the workflow. Once you confirm that the chromatin was properly digested, you go on to stage 3, where you're going to lyse your cells to extract the chromatin fragments that were digested. And then you're going to set up a series of reactions to generate at the end of stage 3, proximity ligated DNA. That's a DNA that you're then going to convert into a sequencing library by completing the last 2 stages of the workflow, stage 4 and 5. So at the end of stage 5, you have a library that you can sequence on any Illumina sequencing platform. So if you're looking to do a technical replication, you're going to start with your sample aliquot. The standard protocol calls for 1 million cells. You can also start with tissue or blood sample. So in this case, I'm just describing the process for a 1 million cell aliquot. So you start with your cell aliquot. You go through stages 1, 2, and 3. And usually to go through stage 3 or the proximity ligation reactions, you're going to input around 1,500 nanogram of chromatin into those reactions. And we expect that at the end of stage 3, when you've completed your reactions, you're going to recover around 50% of that DNA you started with. So in this case, we're going to recover around 750 nanogram of DNA. In order to complete the workflow and generate your library, you only need 150 nanogram aliquot per library conversion. What that means is that from the same proximity ligated DNA and that same proximity ligation reaction, you can take 3 150 nanogram aliquots, convert each into a library, so end up with 3 libraries, and these would be your technical replicates, and then sequence each library to 300 million read pairs, merge the FASTQs, and get your 800 to 900 million read pairs. So your ATX coverage that you need for loop calling. If you're looking for a more biological replication, you're going to start with 3 aliquots of your samples, so 3 pellets of 1 million cells each. You're going to process these through stage 1 to 3. So you're going to do your cross-linking, digestion, lysis, and proximity ligation reactions. You're also going to end up your proximity ligation reactions, or stage 3, with around 750 nanograms, so half what you started with. But in this case, you're only going to take 150 nanogram aliquot from that proximity ligated DNA for each prep. You're going to store the remaining DNA, and then you will convert that aliquot into a library. So really, you have 3 libraries, or 3 biological replicates, generated from 3 proximity ligation reactions that were processed from 3 1 million cell aliquots. Each library is then also sequenced to 300 million read pairs. You merge these FASTQs, and you get your ADX coverage for loop calling. Some researchers do a combination of both, so they will have their biological replication, but from that DNA, they're also going to do their technical replication. And that would be the most robust way of designing your experiment, but this also comes with you having to do more of those libraries. So when you're prepping all of these libraries and you're going to have all these different data sets in your study, one question always pops up is about how much variability am I introducing in my study? How can I reduce that bias when I'm setting up my experiments? And you can do that while you're doing the preparation. So there's definitely a room to reduce that technical source of bias between your replicates and your conditions. So first, by having that same cross-link conditions that are used for each of your sample, in our assay, we do the cross-linking by using formaldehyde and DSG. So it's a 2-step cross-linking method. So you're going to have the same cross-linking conditions between replicates and conditions. You're going to try and aim for that same digestion profile. So I'm showing 2 examples of digestion profiles analyzed on a bioanalyzer. Here we're showing a profile that has 66% mononucleosomes, which are usually represented by that first peak around 150 base pair, which is the size that you would expect your mononucleosomes at. And this is the second profile that shows that the mononucleosomes are around 33%. So both of these profiles would actually pass our QC that you're looking at in stage 2, and you have the opportunity to proceed with these lysates with the rest of the protocol. But if what you're doing is more a comparison study, and both of these samples are part of your same study, we encourage you to aim for a similar profile. So in that sense, you would actually redigest your sample, this sample, to get a more profile that's closer to this sample. So you have the chromatin digested in a more similar way with a similar percentage of mononucleosomes. So this is something you can actually control by aiming for a more similar digestion profile, because digestion profiles do have an impact on library quality. And in our workflow, we make redigestion easy and possible. So if you have a sample that looks like it's less digested than your first sample, you can actually continue your digestion without having to start over. So you just add more MNAs, digest your sample further, and proceed with the rest of your protocol. Another thing that you can control is a ligation reaction. So the way you're setting up your reactions, respecting the time, incubation temperatures, the incubation times as set in the protocol, and using the same thing for all of your samples. And then the library size. So we ask in the last step of the protocol to size select your libraries to a same range between 350 and 1,000 base pair in order for you to proceed with a library that has the same size range for fragments. So these are things that you can control, and you can do so easily by just following the protocol and using the same standardized steps between all of your samples. But regardless of how much work upstream you put into making sure that you're not introducing bias in your experiment, you're going to end up with data that will show a difference. So this is library 1, library 2, and these are just examples of 2 libraries that were generated and sequenced to 300 million read pairs. And you're going to start to see some difference in the data set between percentage of read pairs that map to the genome versus read pairs that do not map to the genome. So there's a slight difference in mappability. You're also going to have a difference in the percentage of read pairs that are actually PCR duplicates versus non-PCR duplicates, so unique read pairs. And then there's a difference in the partitioning of the read pairs between cis and trans. And one level lower, it's the actual partitioning of cis read pairs between those which are less than 1 kb insert size to more than 1 kb insert size. And here we define it as 1 kb because we ask you that you size select your read pairs to 1 kb. So anything that maps with a less than 1 kb, these are fragments that are coming from that same genomic locus. So we refer to as invalid read pairs, things that are more generated with religation events. So you're cutting the fragments and then it ligates in that same position. Read pairs that ligate more than 1 kb, these are your proximity ligated fragments that are coming from ligating fragments that were co-localized in the 3D space. So all of that difference between the partitioning of the read pairs ultimately gets you to one main output that we look at in our data sets, which is what is the number of unique non-duplicate valid read pairs that you have per sample or per replica. And that number ultimately is a result of all the different partitioning that you had upstream. And what we see is that this number will be different or can vary between samples and replicates, despite you setting up your experiment in the same way across all of your study. And this is where I want to hand it over to John to walk you through how do you actually work around having a different number of valid read pairs in your data set. So John, I'll stop sharing from my end and I'll hand it over to you.
Great, thank you, Miriam. Do you see the screen? I think just want to double check everything's good. Perfect. Perfect. So I think what we'd like to do with the rest of this this webinar is a couple of things. I think one, just a brief review of kind of some of the normalization methods that exist. What are their uses, potential limitations, and depending on your experiment, which one you might choose. Also, hopefully simplify that decision-making process. I know there's a lot of tools out there. You see a lot of different methods. At the end of the day, I think we'd like to simplify that process and hopefully, help that decision. I'll show a few analysis examples. Fortunately, there's no catch-all solution for everybody's experiment. But in general, I think most people will fall under one of these three sort of broad, oversimplified categories. Historically, Hi-C data or Hi-C-like data has been done in sort of a single sample way, and mainly because it was so expensive when it first came out and so I guess complex to analyze. A lot of the methods you see in the literature are focused on this single sample within a single sample. How do you normalize the data? But more recently, as the technique has become more accessible and sequencing has become more affordable, we're seeing more and more scenarios where we want to do comparisons between samples, either single sample to single sample or a group of samples, whether they're biological replicates or technical replicates, again, between two samples. So I'll just give a brief overview of some of the tools that are available, when to use them, and hopefully simplify when you would choose one or the other. So to start with, within sample, the large majority of methods you'll see in the literature are within an individual sample. How do we normalize the signal? And they can be, I guess, broadly categorized into, you'll see two main types in the literature: explicit and implicit methods, but more recently we're seeing additional methods coming into play here, which are kind of a hybrid between both of them. So for the explicit, what does explicit normalization mean? And this is one of the first ones that was shown. One of the first methods that was described for normalizing high-C data was this method called high-C norm. And explicit means you need an explicit knowledge of your sample to account for specific variables such as GC content, mappability, or copy number. What it's meaning is you explicitly have to model those parameters. In many cases, we can. We know the GC content of the human genome. We know mappability of the human genome, but when you start bringing in other genomes, or you start accounting for things that you might not have thought about, such as copy number, you might not know what the copy number status of your sample is. The explicit methods is limited because you do need that a priori knowledge, and you run the risk of potentially normalizing out some biological signal. The contrast to that, and what you see most commonly, are these implicit methods. There's just a few of them: vanilla, SCN, ICE, and KR are probably the two that you see the most frequently because they're built into, I guess, the most commonly used analysis platforms, juicer tools, and Hi-C Pro. They all have either ICE or KR, or potentially SCN and vanilla as well built into the analysis pipeline. And what this means is you don't need to know anything really about your sample. It has an overarching assumption that the coverage within each bin that you're normalizing is equally visible. Really, what that means is the limitation of that is you risk potentially overfitting again some biological signal. In particular cases where you have really complex copy numbers, or if you don't know the ploidy of your genome, for example, that assumption can sometimes not be appropriate. Or if you do a capture experiment where you're only capturing certain parts of the genome, these implicit equal coverage type normalization procedures aren't always appropriate. But they do a really good job for the large majority of chromatin topology experiments, where you have a diploid genome and what you're interested in is topology. The ICE and KR method do a really good job. Lately, we've seen others called, I call them hybrid, but really they use some sort of knowledge. They model some features of your genome, and they also borrow on the same concepts that came before them. So binless is a resolution independent one. We haven't worked a lot with it, and I think it'll be interesting to investigate some of these more resolution agnostic approaches. But we're also seeing other approaches pop up specifically for highly complex karyotypes, such as cancer samples like 1D. This one's called CalCB. These ones account for the various different copy number that you can get, or if you have complex karyotypes, or if you outright really don't know if the ploidy of your genome is a relevant factor. These types of tools are really good for those scenarios. So these are, again, within an individual sample, how do we normalize the signal? The contrast to that, and something we're seeing more and more, are these between sample normalization questions. And we are getting a lot of questions of like which ones we do, how do we compare biological replicates to technical replicate? And really, I'd hope to simplify that process a little bit. I think there's basically three ways you can approach a between sample comparison. The first, you notice the box overlaps with the previous one. If you're comparing one sample versus one sample, you can do them individually, or you can do them together as well. And I'll give three examples of when you might choose to do one or the other. But a simple subtraction or fold change, when in the case of comparing one sample to one sample, is oftentimes all you really need to do, especially if you're comparing data set, like your internal data set to the literature, for example. The literature might deposit a list of TADs, and you just want to ask the question, is my TAD is the TAD that I see in this particular cell system that was profiled using a completely different technique, is my TAD in there? And so you may just do a simple subtraction of TAD calls or AB compartment calls, or a fold change between one matrix and another. So these, obviously, the limitation of this is you're going to find everything that's different between your samples. There's not a statistical value attached to that of significance. So you will get a lot of difference. Sensitivity would be basically limitless because you're just calculating what's the difference. The step a little bit more conservative is to take a simple model approach. And this is borrowing just on concepts that are very well known. Anytime you have a matrix of data, which is really, at the end of the day, what we're working with is an interaction matrix, we can apply a lot of the tools that are designed for matrices. Principal component analysis is a dimensionality reduction tool that you can basically reduce your samples into its components and do a comparison of the broad features that are exposed. Or you can use a simple count-based model like a Poisson distribution and really calculate, okay, where are my counts really different between samples? So it's a little bit more conservative than just doing a straight up subtraction or fold change, but you do run the risk of overfitting in the scenario too. What we're seeing pop up more and more are these between sample normalization methods that are actually specifically modeled for a high C-like distribution. And the reason why these are, I don't want to say better, I think they're appropriate in certain contexts, but what the more recent tools such as Hi-C compare, multi-Hi-C compare will do is account for the very skewed distribution at the very close close distance, right? So we know from Hi-C, the closer you are in space, the more likely you are to interact. So the distribution is actually highly, highly skewed towards very small distributions. So when you're doing these more simple Poisson-based models, you're going to skew your results to the very large interactions that are not likely to be interacting with each other. Whereas the more general models, they work a lot better in the smaller space. So if you do want to do a more of a de novo approach, we will lean towards this. And again, the main difference between these types of methods is if you're doing just a simple subtraction, you're less conservative. You're going to find everything that is different between your samples. If you want to get more conservative, that's when we start applying more specific models for our Hi-C data. So I'll just go over a few examples of what these analyses might look like. Everyone's question is different, but I think this covers a number of scenarios. This is just a simple subtraction method. And again, one question might be, is my region of interest in the active or inactive compartment relative to some external data set? It doesn't have to be active or interactive. It could be, a topological domain. I want to know is this domain that I'm working with, this operon in my sample, is it the same in another cell system? So you might want to download some data from ENCODE or wherever you might find relevant data for your sample and just do a simple bed tools, right? Because at the end of the day, TADs and AB compartments are regions, and you can do a simple, region subtraction of, I have sample A, sample B, bed file, I can intersect them and find the unique regions. And I know it seems really simple, but this is oftentimes all you need to do, especially when you're looking at your specific locus. And do a little bit more of a computational approach. You can actually take the matrices and do what's a simple fold change, fold change calculation. So you're not working at the level of the call, you're working, in this case, at the level of the matrix. For example, the cool file. And FANCI is a tool that will do this type of calculation where you calculate simply the difference between fold change between A and B. At the end of the day, what you get is another matrix that has contacts that are enriched in sample A, contacts that are enriched in sample B. Gives you just a really simple way to find where are the differences in the regions. Again, there's no p-value attached to it. There's no significance. But if all you're interested in is your locus of interest, this might be appropriate to you. Or if the data set that you're comparing to is something that's completely external and processed in a completely different way, you might only be interested in the qualitative difference between your samples. In that case, this type of simple approach might be appropriate for that. If you want to do a little bit more of a computational or statistical approach, just going back to our question of the AB compartment difference, one way to do that is with principal component analysis. And this is just one example. FANCI has its own approach, calculating eigenvectors and breaking down AB compartments into its individual blocks. Homer is another tool originally designed for ChIP-seq, but the same principles apply where you can take in your two samples. This is just an example, K4 dimethylation compared to some control sample. And you can perform PCA, find where your blocks of biological signal are really, and compare the principal components to one another. So it's a little bit more of a de novo approach where you're looking kind of genome wide and you want to find where are the big differences between my samples. So this would be considered something in the middle of a simple subtraction with a little bit of borrowing from previous statistical approaches. What we're seeing more often, and I think we're going to continue seeing this, is how to actually do a joint normalization. So what I showed you above with the two examples are when you're normalized the sample internally using either ICE or KR, you take your normalized data and you compare them to each other. But we're seeing more and more now that we can actually do this in a joint method, and there are tools emerging that will perform a Hi-C specific normalization that's appropriate for Hi-C-like distribution. Hi-C compare is just one example. This is an example script. It's not too difficult to incorporate in your own analysis. Even something you can probably plug into an automated type of analysis. This is where you take at the matrix level, 2 matrices. There's a little bit of pre-processing that extracts the counts. Because the main difference now, because we're doing joint normalization, is that we're using the raw counts as opposed to the ICE or KR normalized data. So we can dump the counts, and there's a normalization step where both counts are jointly normalized using their model, which is a loss-based GLM model. And you can output basically a list of differential regions with a p-value attached. It's nice because you can do this one-to-one, individual sample to individual sample. If you're so lucky that you have replicates, genome-wide, micro-C or omni-C replicates or other Hi-C, you can also do this in a group-to-group fashion as well. This is what the output of that might look like. Normalization step, again, is the main difference between, I guess, the methods that are coming out are they account for the difference in distribution that is a function of distance. All right, so this is, you've probably seen plots like this before. This is a loss curve, which is a function of interaction frequency against basically distance, and it normalizes for that scenario. At the end of the day, what you get is something really similar to what you see for an RNA-seq experiment, where you can compute, volcano plots, see what are the regions of interest specific to one sample or one group of samples to another. And this is a nice example of what the output of that might be. A list of regions that are differentially interacting with each other, and we pull up those regions and we see there's loops that are specific to one or the other, complete entire topological domains that are enriched in one sample or another. So we're seeing a lot of this, and I think as cost of sequencing continues to go down and the accessibility of these techniques, I think we're going to see more of these methods as well. There are emerging methods. 1D, like I said, is something appropriate when you have highly complex cancer samples. We haven't performed these internally yet, but they do seem to do a good job of accounting for specific scenarios. So this one is if you have like I said, cancer samples that have really messed up karyotypes, or you have 6N chromosomes and you have 1N chromosomes, this adjusts for that copy number difference without removing that biological signal. Another one, binless, is resolution agnostic. So what we're talking about are methods historically have been done at certain resolutions. You can choose the resolution that you want to perform your analysis. Binless supposedly is a way to do that without actually caring about resolution. So you can do an normalization up front, and then you can perform your analysis downstream afterwards. So everything I've been talking about has really been on the micro-C side, or the omni-C, which is whole genome. But what do you do if there's a capture? A lot of the tools that you'll see developed with historical Hi-C data are meant for these genome-wide analyses. We do have 2 types of capture. One is the pan-promoter capture, another is called HiChip, which really is an addition of a hybridization or some sort of selection step. So when you do that selection, a lot of the tools that were previously developed, like ICE and KR, their assumption kind of goes out the window, because now you're looking at a very small region. The even coverage across the genome no longer applies. Thankfully, there are tools available that specifically account for the specific model that that we see for this type of data. So Chicago is our method of choice for capture data, specifically designed for capture, and conveniently comes with a differential pipeline downstream as well. And the same is for fit high chip. So high chip data is where you do an antibody selection. Fit high chip is designed to specifically handle that type of data. We'll call loops, and also comes packaged with a downstream normalization technique, or sorry, a differential analysis, which also includes normalization. So there are solutions available for these specialized scenarios as well. So this is a cheat sheet that hopefully will simplify that process. I don't think we need to go over every word right now, but basically, depending on what your goal is, if you're looking within individual samples and you want to explore your data, ICE and KR do just fine. And these are packaged with the majority of tools that are designed for processing high-C data. When you start comparing to other biological systems, we often can just use a simple comparison, a subtraction, a simple Poisson-based model. When we start introducing biological conditions, say, and as you're designing your experiment, you're probably designing, as Miriam alluded to, as like a wild type versus a knockout, or some sort of cancer A versus a control. Then we start bringing in a joint normalization, because we want to do a specific genome-wide de novo comparison between conditions that were profiled in the same way. So high-C compare, multi-high-C compare handle that type of analysis. And there are targeted loci specific capture, like pan promoter, high chip. There are tools specifically designed for handling that type of data. And I alluded to, I think one that is common is we're starting to do this in individual cancer samples, and there are tools that are available that will hopefully account for those complex samples. Just wanted to end off on one sort of practical consideration. It's a question that we get a lot, which is, okay, if there's these two different types of normalization methods, there's within sample, there's between sample. We frequently get the question is, should I normalize within sample first and then do a between sample? And thankfully, the format of the high-C, the two main high-C file formats, cool and high-C, what they do is they store the within sample normalization as an internal weight. It's just an additional column that's added to the raw data. So even in the high-C and cool files that have gone through ICE or KR normalization, or any other type of within sample normalization, that normalization parameter gets stored internally, which is very convenient, because when you start introducing between sample analyses, what they want are the raw counts, because they're doing their own statistical modeling directly from the raw data. And that's still available. And you'll see the majority of these tools, what they'll do is they'll take the cool file and they'll do some sort of pre-processing, they'll extract the raw data. And so you can still do the joint normalization. So what that means practically is when you're designing a pipeline, you can go ahead, do your within sample normalization. That's the file that you're going to use for your visualizations when you're streaming to, for example, juicer tools or some sort of browser. You'll want that within sample normalized value. And then you can still do the raw data between sample normalizations directly from that. So you don't have to worry about having two different pipelines with two different normalization strategies. Thankfully, it's already built in. So everything that I spoke about is in a little bit more detail with the in a recent blog post that we have. I'll just put the link here. I think we're going to circulate these slides. You don't have to write it down, but we talk about this in a blog post. And as well, everything that we've spoken about is available as a pipeline that we've set up. And it's hard to catch all, but we do have simple condition A, condition B for biological, for technical replicates, for single samples. We do have a service available for doing that, and even comes with downstream annotations. Once we've done your comparison, and we identify these regions of interest, we can do lots of fun stuff, and we offer a pipeline service for that type of thing as well. So I think that does it for me, and I see there's a lot of questions, so I think we should get to the questions there.
Thank you, Jonathan. Miriam, did you want to read out the questions, or I can go ahead and do that?
You can go ahead, Jesse, if you don't mind.
Yeah, why don't I do that so that you can answer them too. Okay, starting with number one, ignore this if it's covered, but are there any procedures that are recommended for scaffolding polyploid genomes?
Okay, so yes, we do offer a solution, especially for polyploid genomes. And I think what would be best here is if I connect you with our sales rep who can walk you through all the different solutions that we have. We offer this currently as a service. So following this webinar, I'm going to just connect you with our sales rep who can talk you through what we have for polyploid genomes.
Okay, thanks. Next question is, would both technical and biological replicates generate the same sequencing results for loop at the 1 kb resolution?
So for 1 kb resolution, I think I covered mostly until 5 kb resolution. If you're looking for higher resolutions like 1 kb, the coverage that you're after is around 200x coverage. And that is accomplished by getting to around 2 billion read pairs. So the 2 billion read pairs, you would need around 7 libraries to support that depth. And a lot of the times what I see when it's that depth that is needed or that number of libraries that is needed, researchers do a sort of a combination between technical and biological replication. And I will share my slide. I added one slide to talk about combination of biological and technical replication. So here we go. So this will allow you to achieve the number of libraries that you need and the depths that you need for a 1 kb resolution and higher when you're calling loops. And here, what you're really doing is you're starting with 3 million cell aliquots. For each preparation, you're going through stage 1, 2, and 3, finishing your stage 3 or proximity ligation reactions with around 750 nanogram yield for each of these preps. And then you're going to take 150 nanogram aliquot, but 3 times. So 3 aliquots from your proximity ligated DNA. And that's again for each preparation. And you're going to convert each of those 150 nanogram aliquot into a library. So essentially what you're generating is you're generating 9 libraries from 3 proximity ligation reactions and 3 1 million cell aliquots. And then each of those 9 libraries is going to be sequenced to 300 million read pairs, keeping in mind that that's the highest depth that is supported by a library. And then you're going to merge these sequences together. And that will give you around 2.7 billion read pairs. So more than what you need for a 1 kb resolution and even higher. And in that way, what you can do is you can compare. So you have your data set to merge all together and do that loop calling at 1 kb resolution, but you also can merge them at 900 million read pairs if you want to compare between the samples. So that's mostly what I've seen researchers do if they want to make the most out of their reactions, and if they don't have a lot of cell availability or sample availability to do, endless number of biological replicates. That would be one way to achieve 1 kb resolution. I hope this answers the question. If not, feel free to just let me know if you have any further questions on that.
Okay, thanks, Miriam. Next question. My purpose to use Hi-C data is helping scaffold genome assembly. What coverage of genome size do I need to achieve this? In my case, the sequences assembled in my PacBio Hi-Fi assembled genome. But it didn't close any gap. I need more data. Do I need more data from the same library, or do I need to prepare another library? Thank you.
So here the Hi-C data would be perfect for allowing you to scaffold an assembly that was generated with PacBio. Usually, what we would aim for scaffolding is a 30x coverage of your genome. So that's, the depth that you need is obviously dependent on your genome size. So if we're thinking about 1 gigabase genome, to achieve 30x coverage, you need around 100 million read pairs. And that's a 2 by 150 base pair read length. So if you're looking at 3 gigabase genome, you need 300 million read pairs that can be achieved with one library. if your genome size is, for example, 6 gigabase, you need 600 million read pairs for 30x coverage, and then you need 2 libraries to achieve that. And these are libraries that you need to generate from the solution that we offer for genome scaffolding is called OmniC, which follows the same kind of workflow that MicroC, which we were talking about in this webinar, but uses a DNase for digesting the chromatin instead of an MNase, which is used in MicroC. And the use of DNase allow you to get a more uniform coverage of your genome, which is really helpful for closing gaps and scaffolding your genome for genome assembly application. The kits we were talking about in today's webinar are more based on MNase digestion of the chromatin, which will allow you to achieve the highest possible resolution for any epigenetic studies.
Okay, thank you. If I do replicates from stage 3, the ligation stage, would it be more advantageous in comparison to the technical replicates to reach 1 kb loop resolution?
I think this we addressed with the previous question as well.
Yeah, it sounds like it actually. Yeah.
For 1 kb resolution, you need to get to 200x coverage, 2 billion read pairs. Different ways of doing this, but one way you would want to consider is combination of technical and biological replication in your experimental design.
Yep. And someone just asked, what's the follow-up email? I'm asking for the quote of high chip instead of high C kits. Why don't we, Chen Guang, why don't we connect you with one of our sales reps that can help you with that? The follow-up email that I'm sending out is the recording as well as the slides. But when I do send that email out, you can respond and we can connect you with somebody. Additionally, there's a form on our website that you can easily fill out and they will get right back to you for quote requests. Okay, next question. Can you give some advice as to how we can adjust these steps or view our data in the context of promoter capture high C data?
I think this one, John, do you want to take that on?
Sure. So I guess if you're talking about the computational steps, I think when we're talking about the capture data, you probably want to use a tool that's designed to account for the distribution that you'll get for a capture experiment. So we showed you 2 examples of that. Doing target capture, there's a tool called Chicago, which will specifically perform a model-based normalization that's specific for the distribution of counts that you get for capture, and the same for high chip. The steps from a practical perspective are the same. You do your experiment, you'll get your reads, you'll generate your pairs file, and then the tools downstream handle it from that perspective. So you won't necessarily need to do like an ICE or KR normalization. You can take the data directly, plug it into a tool designed for that. And yeah, the tools practically are the same. I hope that answers your question. I'm not sure if it was a wet lab question, but yeah, there's tools that will account for that.
Okay, thank you. Next question is in 2 parts. And the first part, is it acceptable or proper to do differential analysis without duplicates? Part 2 is, what's the common use of FTR value for loop TAD calls? Less than 0.2, 0.1, or 0.05?
Yeah, thank you for that question. I think it's a question that we get a lot as well. And the short answer, and it might not be a satisfying answer, but the differential literature is far behind, I think, what you see in the RNA-seq world. The RNA-seq world, there's a lot of truth set data that you can, and now you can ask the question, how robust is my data relative to a gold standard that we know a lot about? For high C, that sort of gold standard doesn't really exist. So to answer the question, acceptable or proper is really sort of I don't want to say an arbitrary decision, but we don't know what the truth is really. To ask whether the calls we get at 0.2 or 1 or 0.5 are the true calls or not. The nice thing is when you do this analysis, it gives you a short list of, I think, what are the best regions in your sample. And you might have to go down 1,000 regions out of 1,000,000 regions to get the one that you're interested in or the hypothesis that you want to test. But there's no real answer to what is my cutoff, where is the biological signal end. I think at the end of the day, we have to consider that these methods are really hypothesis generating methods, where we want to know what are the differences between samples, the best that we can do. And then you usually have to do some sort of downstream validation or experiment. And that's where Go annotation comes in. Are the results making sense is really a relative question depending on what type of system you're using. So there's no black and white cutoff, but what the tools do is they point you in the right direction. They'll tell you these are the most statistically robust differences between my samples. And oftentimes the nice thing is it's hard to fake TAD signal, right? If you look at a TAD, it's very distinct what a TAD looks like in your region. So if you pull both samples up and there's a TAD in one and there's not a TAD in the other, that to me is the validation. That's, you can't fake that. So I know it's not a satisfying answer. It's proper. You can do one-to-one, you can do group-to-group. The result will be valid and proper depending on kind of what your hypothesis and what your what your goal is. So I wouldn't worry. I think that at the end of the day, don't worry. I think, the tools are going to generate hypotheses. And I think you're going to find with a lot of the top differential regions that you're going to find is going to be a true biological signal.
Okay, great. Next question is, what is the difference between the raw Hi-C data and the ICE normalized Hi-C data? And then which one should we use when we try to compare two Hi-C maps?
Yeah, so the nice thing is the difference is an additional column in your Hi-C or your cool file. So when you have a Hi-C or cool file, that's your interaction matrix at multiple resolutions. When you do a normalization, such as ICE, all that does is it adds a weight column. All right, so now it's assigning a normalization weight for each region, and that weight is used downstream by like juicer tools or any of the visualization tools. What they do is they calculate that normalization on the fly. So practically what that means is you're not changing any of the data. The Hi-C and the cool file that has the raw data, and then every time you do a normalization, it will add a normalization weight to it. So when you're just pulling up and you're viewing two Hi-C maps, as long as you've done some normalization, I think that's what you're gonna see is the normalized weight calculated on the fly. When you're going downstream and you're doing more of a differential analysis where you want to pull out all the regions that are different, that's gonna take in the raw counts directly from the file and generate the statistics based on that. So the real answer is there's no difference between them other than when you normalize the data, it adds a normalization weight to the data.
Okay, thank you. Let's see. Next question is, I've been working with Hi-C Explorer, and for comparing two samples, they recommend correcting the matrix first. Let's see, with KR, VC, squirt, I'm just going to say it like that, etc. And then normalizing the matrix after the smallest read depth, the normalization range, etc. Do you think this is too conservative and that one of these would suffice? Do they only need to use one of them?
Yeah, I'd have to read through the specifics of what they're recommending, but the correcting part, to me that's normalization. That's correcting for your within sample Hi-C specific bias, right? So the KR, the V square root, the ICE, those are the within sample corrections, or I refer to that as just within sample normalization. I'm not sure what normalizing the matrix after means, but I think the joint normalization tools are the ones that are going to be doing that, read depth and count based normalization. But this is something I can read into what Hi-C Explorer is recommending. I don't know off the top of my head what exactly they're recommending, but in general, what you can do is do a nice KR within normalization. That's more than valid for doing single sample comparisons within a single sample. And when you start comparing between samples, that's when you want to use a joint normalization, which at the end of the day is going to go for the raw data anyway. So it doesn't want a normalized matrix. It wants the raw counts when you start comparing between samples. But yeah, I will look into what the Hi-C Explorer recommendation is as well.
Okay, thank you. Next question is, how are the two Hi-C maps, liver and brain, normalized in the example that was shown?
Yep, so that would be KR. That's our default, just because it seems to work faster. And you'll find a number of comparisons of like ICE versus KR in the literature, and they're very similar. There's only minor differences. So we use KR for speed purposes. And then for Hi-C compare, so that analysis was done using Hi-C compare, which is a joint normalization that basically extracts the raw data and does the joint modeling. So yeah, KR for the figures and raw joint normalization for getting the p-values in the regions.
Okay. We've got several more questions. Let's do a few more, and then we can, if we run out of time, we're almost at the hour, then we can answer your questions over email. So don't worry, we will get to all your questions. If we don't do it live, we will send you an email. So the next question, what if I have two compared groups and each of the group contains multiple samples? In this case, if I want to identify the true chromatin interactions, which software normalization should or can I use?
Yep, so our recommendation for one-to-one, so that's one sample to one sample, is Hi-C compare. And conveniently for the group to group is multi-Hi-C compare. So that's our preference. Seems to work well in-house. There are others emerging, but those are the two that we recommend right now.
Okay, great. And next one is, when we run the pipeline, it will generate lots of intermediate files. Many of them are very large size and have to be deleted. Could you please let us know what type of files we should always keep in case we need for future analysis?
Yeah, I don't want to answer with like a catch-all solution, but for sure you'll want some sort of matrix file, either the Hi-C or the cool file, depending on whether you're using cooler tools or whether you're using Juicer, Hi-C Explorer. Everything else is depending on your experiments. So some downstream, if you're in cancer research, for example, the BAM file that's generated, like the valid pairs BAM file, is also often useful for doing like variant calling. But for sure, the Hi-C file, you'll probably want to keep, depending on how you're calculating your unique pair statistics. We use pair tools that generates like a statistic file. And it's hard to answer that with a catch-all, but for sure, the matrix file is pretty much all you're going to need for downstream analysis, either the .Hi-C or the .Cool.
Okay, great. Next one is, while scaffolding our genome of interest using Hi-C reads, we detected misassemblies. Are there ways to solve these misassemblies? Is normalization a way to go?
Sure, I guess I can take that one. So the normalization that we've been speaking about is really in the world of chromatin topology. I don't know, Miriam, this might be more of a specific question that we could answer offline, but the normalizations that we use, you probably don't want to apply them for genome assembly, for scaffolding. But yeah, I think that's potentially something we can refer you to offline.
Yeah, so there is a way, as John mentioned, it's not really the normalization that's applicable here. Our team does do a manual correction, which other teams do as well when they detect misassemblies. So after you have your outpost file from the scaffolding pipelines, you would still need to do some manual corrections for some of those genomes. But we'll connect you with our team who are experts on genome scaffolding.
Okay, thanks.
And I think that will probably apply to the next question as well, which is
okay.
The normalization for the genome assembly world is a little bit of a different thing. So probably there's better experts to talk about like the specifics between how do we handle those cases.
Okay, then we'll get in touch with you directly with answers to those questions regarding genome assembly. So we have several more, and we're at the top of the hour now, and I think both of you probably have meetings at the next in the next hour. So why don't we take the rest of the questions offline, and we'll answer those over email. Don't worry, we have all of your email addresses from when you registered for the webinar, so we'll make sure to get through these questions and send you out responses just as soon as we are able. Thank you so much, everyone, for joining us today. It was a really wonderful discussion, great presentations, and we really appreciate you being here and asking these questions. It really helps everybody in the community. So I'm very grateful for that. Thanks so much, speakers, and we look forward to seeing you guys at the next webinar. Take care for now. Bye-bye.